Welcome back, everybody, for another deep dive. This time we're diving deep into the world of DNA,
Oh, yeah?
specifically a technique called PCR.
Right.
Now, you can think of PCR kind of like a DNA photocopier,
Okay.
and I'm not even exaggerating when I say this thing revolutionized everything we know about genetics.
Hmm.
Before PCR, studying DNA was like trying to read a 6 billion-letter book without a magnifying glass.
Right.
I mean, you could technically extract DNA, like it's surprisingly easy to do, even with some basic household items,
Oh, wow.
but you couldn't really do anything with it.
That's right. Even with a really high-powered microscope, raw DNA just looks like a shapeless blob.
Oh, really?
We can't make out individual genes or mutations or anything useful, really.
So it's kind of like having all the ingredients for a cake, but no recipe and no oven.
Exactly. You can't bake anything.
So we had all this DNA, this incredible blueprint of life, but we couldn't really decipher it.
Exactly. And it's not like we were dealing with a small instruction manual either.
Right.
We're talking about 6 billion base pairs. The individual building blocks of DNA crammed into every single cell.
That's a lot.
And early techniques were really slow and limited.
I bet.
Scientists were using things like restriction enzymes to cut DNA at specific points.
Okay.
But even finding a tiny mutation like the one that causes sickle cell anemia was a huge challenge back then.
Yeah, I remember reading about the Southern blot technique,
Right.
which used radioactive materials to identify specific DNA sequences.
Yeah,
Sounds incredibly time-consuming and a little bit scary, to be honest.
it was. And that's really what makes the invention of PCR so revolutionary.
Okay.
It's like going from reading by candlelight to having a high-powered spotlight that illuminates exactly what you need to see.
Oh, wow. So who came up with this revolutionary technique?
That would be Kary Mullis, a scientist with a personality as colorful as the DNA strands he was studying.
Interesting.
Instead of trying to zoom in on specific DNA sequences,
Right.
he imagined making billions of copies of the DNA he needed,
Yeah.
basically amplifying it until it was easy to see and analyze.
Okay. That sounds incredibly useful, but also super complicated.
Yeah.
How did he even begin to figure that out?
Well, the story goes that he had his eureka moment while driving to his cabin one night in 1983.
While he was driving.
Yeah. He envisioned a process that mimicked the way DNA replicates naturally in our cells. Basically, you heat up the DNA to separate the two strands.
Okay.
Attach little pieces of DNA called primers to the target sequence, and then use an enzyme called DNA polymerase to copy the sequence between those primers.
So you're copying just a specific section.
Yeah, rinse and repeat that cycle about 30 times, and boom, you've got over 1 billion copies of your target DNA.
Wait, so it's kind of like a copy machine, but for DNA.
That's a great way to put it.
Yeah.
And the beauty of PCR is its simplicity and elegance.
I like that.
But of course, no scientific breakthrough comes without its share of challenges and doubters.
Right. From what I've read about Mullis, he wasn't exactly known for playing by the rules.
Haha. No, he wasn't.
I even heard there were some fights in the lab.
Oh, yeah. He was a very passionate scientist, let's just say that.
Sounds like my kind of scientist. But seriously, how did people react to his idea?
Well, his colleagues at Cetus Corporation, an early biotech company, were skeptical at first.
Really?
Yeah, they thought the idea was too simple to work.
Huh?
And to be fair to them, the initial experiments were far from perfect.
So even a genius like Mullis hit a few roadblocks.
Oh, yeah, of course.
What was the problem?
Well, the problem was the high temperatures that were needed to separate the DNA strands.
Right.
It kept destroying the DNA polymerase enzyme,
Oh.
which was essential for making those copies.
I see.
It was like trying to bake a cake in an oven that's way too hot.
Yeah.
You just end up with a burnt mess.
So they needed an enzyme that could, like, handle the heat?
Exactly.
But where do you even find something like that?
Believe it or not, the answer was found in a hot spring
Really?
at Yellowstone National Park.
Wait, seriously? Like in those colorful steaming pools that tourists flock to?
Exactly. Those ones. Turns out there's a bacterium called Thermus aquaticus,
What a name.
or TAC for short,
Okay.
and lives in those boiling waters.
It can survive that.
Yeah, quite happily, in fact.
Oh, wow.
And even more amazingly, it produces an enzyme
Does it really?
called TAC polymerase
Okay.
that can withstand those extreme temperatures.
So this little bacterium living in what's essentially a giant hot tub
Right.
held the key to unlocking the full potential of PCR.
It really did.
That's amazing.
It is. Scientists at Cetus, they extracted that TAC polymerase
Uh-huh.
and used it in PCR.
Okay.
And suddenly the process became incredibly efficient and automated.
Oh, wow.
What used to take days or weeks could now be done in just a few hours with incredible accuracy.
So Taq polymerase was like the missing ingredient in the DNA copying recipe.
It was.
That's so cool. And what kind of impact did this breakthrough have?
Well, it's no exaggeration to say that PCR powered by Taq polymerase
Yeah.
revolutionized countless fields.
Like what?
Well, think about forensic science.
Yep.
Before PCR, investigators relied mainly on fingerprints and eyewitness accounts,
Right.
which, let's be honest, aren't always reliable.
Yeah. I've seen enough true crime documentaries to know that eyewitness testimony can be pretty shaky.
Exactly. But PCR made DNA profiling possible, allowing investigators to link suspects to crime scenes with a level of accuracy
Right.
that was unheard of before.
Yeah.
And it's not just about catching criminals either.
What else?
PCR's also been instrumental in overturning wrongful convictions,
Oh, really?
bringing justice to people who were wrongly accused.
Wow.
Just imagine the impact on someone's life,
Yeah.
being exonerated after years in prison thanks to DNA evidence.
It's incredible to think that this tiny bacterium from Yellowstone played a role in righting those wrongs.
I know, it's amazing.
It's like a real-life superhero origin story.
It is. And the impact goes way beyond crime scenes.
Where else?
PCR has also transformed the field of medical diagnostics.
Oh, okay.
Before PCR, diagnosing genetic disorders or detecting viral infections was a long and often uncertain process.
Yeah, I remember reading about the early days of HIV testing.
Right.
It was a terrifying waiting game for people.
It was PCR changed all that.
How so?
It allowed for the rapid and accurate detection of all sorts of conditions,
Oh, wow.
from HIV to cancer to genetic disorders.
That's incredible.
And the speed and accuracy of these diagnoses are absolutely crucial for patient care.
I can imagine.
It's the difference between starting treatment early or catching a disease before it progresses too far.
Right.
You know,
Yeah.
it's literally a lifesaver.
Wow, so PCR is like a detective and a doctor all rolled into one.
Uh-huh. You could say that.
What other fields did it revolutionize?
Well, how about taking a trip back in time?
Okay.
PCR opened up entirely new possibilities for ancient DNA research and evolutionary studies.
Really?
Yeah. We can now analyze DNA extracted from ancient fossils.
Like dinosaur bones and stuff?
Yeah. Even extinct species.
Yeah.
It's like having a window into the past, allowing us to understand the history of life on Earth in ways that we never could before.
That's mind-blowing.
I know, right?
It's like those Jurassic Park movies, but in real life.
Uh-huh. Exactly. And let's not forget about the impact on molecular biology and genetic engineering,
Right.
gene cloning, vaccine development, even the creation of genetically modified organisms.
Wow.
All of these fields rely heavily on PCR. It's really become a cornerstone of modern biotechnology.
It sounds like PCR has touched almost every aspect of our lives in some way.
It really has.
From solving crimes to diagnosing diseases to understanding our ancient past, it's everywhere.
It really is. But like any powerful tool, it also raises some ethical consideration.
Right. It makes you think about the responsibility that comes with such incredible scientific power.
Yeah.
And speaking of responsibility, the story of Kary Mullis, the man behind PCR,
Right.
it's as fascinating as it is complex. He was brilliant, unconventional, and even a little bit controversial.
Yeah.
I mean, he openly credited his use of LSD with helping him come up with the idea for PCR.
He did. He was definitely a unique character.
Yeah.
And while he undoubtedly deserves credit for envisioning PCR,
Right.
it is important to remember that it was a team effort.
Oh, yeah.
Many other scientists at CETUS played crucial roles in refining and developing the technique into what it is today.
So it was more like a scientific relay race.
Yeah.
With Mullis running the first leg and then passing the baton to others.
It's a great analogy.
Right.
But despite the collaborative nature of the work,
Right.
Mullis received sole credit for the Nobel Prize in 1993, which caused quite a bit of controversy in the scientific community.
That makes sense. Science is rarely a solo endeavor.
Right.
But beyond the Nobel Prize drama, Mullis was known for some pretty out-there views. He was a vocal critic of climate change and even denied that HIV caused AIDS,
Yeah,
which is a dangerous and harmful stance to take.
it is. His personal beliefs were often at odds with scientific consensus.
Right.
And unfortunately, those beliefs sometimes had negative consequences,
I see.
especially in the case of HIV and AIDS.
Yeah.
It's a reminder that even brilliant minds can hold deeply flawed beliefs.
It's a good reminder that we should always critically evaluate information,
Yeah,
even if it comes from someone with impressive credentials.
absolutely.
But putting aside his personal views,
Right.
Mullis's invention of PCR really highlights the power of creative thinking
Uh-huh.
and a willingness to challenge conventional wisdom.
Absolutely.
Yeah.
And it's also fascinating to think about the role that automation played in this discovery.
Yeah.
Remember, his job at CETUS was to synthesize DNA,
Right.
which was a tedious and repetitive task.
Yeah.
It was the automation of that process that really freed up his mind to wander and ultimately led to that eureka moment with PCR.
So in a way, we can thank automation for giving Mullis the mental space to stumble upon one of the most significant scientific discoveries of the 20th century.
There's definitely a connection there.
Huh.
And as artificial intelligence and automation technologies continue to advance, it makes you wonder what other groundbreaking discoveries are waiting to be made.
Yeah.
Maybe the next PCR won't come from a lab filled with bubbling beakers and test tubes,
Right.
but from a collaboration between a scientist and a powerful AI.
Interesting.
Crunching massive amounts of data and uncovering patterns we could never have imagined on our own.
It's mind-boggling to think about the possibilities. So for everyone listening, if PCR, a technique born from a late-night drive, and a little bacterium from a hot spring, could revolutionize so many fields, what other world-changing discoveries might be out there, just waiting to be unlocked?
Maybe the next big breakthrough will come from a simple observation in nature. A chance encounter in a lab.
Yeah?
Or even a dream.
Wow.
The key is to stay curious.
Right.
Keep exploring.
Yeah.
And never underestimate the power of human ingenuity, even when it comes from unexpected places.
And on that note of endless possibility, we'll wrap up our deep dive into the incredible world of PCR. We hope you've enjoyed this journey into the heart of DNA, the power of scientific discovery.
Yeah.
Keep those minds curious, and we'll catch you on our next adventure.